I'm going to be talking about phospholipids. Phospholipids are part of the phospholipid bilayer, which surrounds, it's part of the cell membrane of all cells. There's two types of phospholipids. There's glycerophospholipids and sphingophospholipids, and they are different based on their backbones. So glycerophospholipids have glycerol backbones. Sphingophospholipids have sphingosine backbones. Let's come over here and focus on glycerophospholipids. So glycerol backbone with a saturated fatty acid on carbon 1, an unsaturated fatty acid on carbon 2, a phosphate on carbon 3, and a constituent, whether it be inositol, serine, ethanolamine, and choline, and I remember it as I-sec or 1-sec. So phosphatidic acid, so whenever you put inositol as the constituent, the name of the glycerophospholipid will be called phosphatidyl inositol. If you put serine as the constituent, it'll be phosphatidyl serine, and so on. So, but phosphatidic acid itself is this structure. So the glycerol backbone, sat fatty acid, unsat fatty acid, and the phosphate. No constituent. So if we add inositol as the constituent, we get phosphatidyl inositol. Phosphatidyl inositol turns into IP3 and DAG, which are second messengers inside of cells. The carbon 2 on phosphatidyl inositol will have arachidonic acid, and we cut it off using phospholipase A2, which we'll talk about in a second. We cut off arachidonic acid to make eicosanoids, and some eicosanoids are thromboxane, prostaglandins, and leukotrienes. Another glycerophospholipid is phosphatidylcholine. It's also called a lecithin. Phosphatidylcholine is the most abundant glycerophospholipid in our body. It's also a lung surfactant. So if we don't have enough phosphatidylcholine, this is seen in babies a lot. They'll have respiratory distress syndrome. So they won't be able to breathe. Phosphatidylcholine, I always think of acetylcholine, which makes me remember that phosphatidylcholine is part of the plasma membrane in our nervous system. And the carbon 2 of phosphatidylcholine has oleic acid, omega-3 or omega-6, which is turned into arachidonic acid. So it looks like phosphatidylcholine's carbon 2 is, it has oleic, omega-3 or omega-6, and then it's cut off to make arachidonic acid, and then arachidonic acid is on the carbon 2 of phosphatidyl inositol, and that's taken off to make eicosanoids. Phosphatidylserine and ethanolamine are known as cephalins. Whenever I think of cephalins, I think of encephalopathy, so I think of the central nervous system. So it's part of the cell membrane in the brain and spinal cord. Cardiolipins, so cardio makes you think of the heart, so it's PG and DPG, or phosphatidylglycerol and diphosphatidylglycerol. They're part of the cell membrane in our cardiac myocytes. Plasmalogen is another special glycerophospholipid that fights oxidative stress in the brain. Platelet activating factor mediates inflammation in anaphylaxis, and it helps platelets. It's exactly what it sounds like. It activates platelets to aggregate and help form a clot. There's 3 lung surfactants, phosphatidylcholine, and then the cardiolipins, PG and DPG. So the lecithin and cardiolipins are the surfactants. Over here, so sphingophospholipids have 3 little subcategories. There's ceramides, sphingomyelins, and glycosphingolipids. Ceramides, so it's just a sphingosine backbone with a fatty acid and a hydrogen constituent. Ceramides are common in skincare. It's the simplest form of a sphingophospholipid. A sphingomyelin, myelin makes me think of myelin sheath, and this is the phospholipid that makes up the myelin sheath. So it's a sphingosine with a fatty acid and a phosphatidylcholine as the constituent. If you don't have an enzyme called sphingomyelinase, you can't break down sphingomyelin. They'll build up in the tissue and cause something called Niemann-Pick disease. Glycosphingolipids, so glyco is sugar. So it's a sphingolipid with sugars. Cerebroside means there's 1 sugar constituent. Globoside means there's 2 or more sugar constituents. And ganglioside means there's 2 or more sugars and salicylic acid. If you have trouble breaking down cerebrosides, you can have Fabry's or Gaucher's disease. If you have trouble breaking up gangliosides, you may have Tay-Sachs disease. Phospholipids can be degraded using phospholipase enzymes. So PLA1 breaks down the saturated fatty acid off carbon 1. Phospholipase A2 cleaves the unsaturated fatty acid off carbon 2. Phospholipase C cleaves the phosphate on carbon 3. Phospholipase D cleaves the constituent. And there's a disease called sphingolipidosis. That just means that you can't break down sphingolipids. And this is the synthesis pathway. It's similar to tag synthesis, where it starts with glycerol and DHAP turning into G3P and then phosphatidic acid. And you can see that I included tag synthesis going this way, but the rest of phospholipid synthesis goes this way. So if you start with phosphatidic acid, you can add a molecule called CDP, and that'll turn phosphatidic acid into CDP DAG. Or if you get phosphatidic acid and go to DAG, just add a CDP, and then you get CDP DAG that way. But either way, you end up with CDP DAG. If you add inositol to CDP DAG, you get phosphatidylinositol. You can also turn CDP DAG into phosphatidylglycerol, and then phosphatidylglycerol turns into diphosphatidylglycerol. CDP DAG, if you add serine, it'll turn into phosphatidylserine. So that's why I say 1 sec, because it kind of goes in that order. Phosphatidylserine can turn into ethanolamine by switching serine for ethanolamine. You can also go backwards and remove serine and put ethanolamine. Phosphatidylcholine can be made from phosphatidylethylamine, or it can be made by starting with choline, adding a phosphate and getting phosphocholine, adding CDP to get CDP DAG, and then getting phosphatidylcholine. I need to add an arrow right here really quick. You can get phosphatidylethynolamine by starting over here too. You can get ethanolamine, add a phosphate to get phosphoethanolamine, add CDP to get CDP DAG ethanolamine, and then you just get phosphatidylethynolamine. I hope that made sense.